Thank you very much, everyone, for taking time out from this, lunch hour, and hope I can do justice to this very broad topic of chimeric receptor antigen T cell therapies. Really, this is an evolving field and a field that has caused much excitement and has enabled the fast growth of cell tissue and gene therapies over the past 5 to 6 years in various countries across the world. So in this lecture series, as Estella has mentioned, we are talking about very basic concepts. So pardon me for some of you who are out there who are very, very knowledgeable in this field, that I will touch upon very basic concepts to give our audience a broader perspective. With that, I'll just bring you through the outline of this presentation. So at the beginning, I'll talk about some basics of CAR T cells, giving you an overview of some of the products, how it came about, the basic studies, and what are the clinical hurdles and complications along with it. Then move on to talk about some of the recent scientific papers that have been published trying to address some of the key challenges in this field. Moving on to manufacturing processes. And then I wanted to take some time to talk about regulation, but it is such a broad topic that I thought that it would not fit the time frame that we have for this presentation. Therefore, and since this is a education platform, a webinar series hosted by ACTRIS, I wanted to end off by giving our audience an overview of Singapore's cell, tissue, and gene therapy ecosystem, and as well as giving you a bit of an overview of what ACTRIS is and what we are doing together with various other partners across the ecosystem to bring cell, tissue, and gene therapy products to patients. So in terms of the basics, let's get started off with the very core basics of cell, tissue, and gene therapies, right? So here, as you can see, let me get my pointer out. As you can see here, there are three main broad classes of therapies that we are talking about: gene therapy, cell-based immunotherapy, and of course, stem cell-based therapies that are mostly targeted towards regenerative medicine, right? In gene therapy, what is done in a classical gene therapy is that a plasmid through a viral vector is introduced to a patient to exert a certain therapeutic effect, right? And in a cell-based immunotherapy, what we do is that we use similar viral particles with plasmids of interest and then use these to manufacture T cells or various other immune cells that are engineered to express certain markers or certain therapeutic targets that can be used to develop the therapeutics and be given to patients. And cell-based immunotherapies, as of now, has primarily been targeted towards immuno-oncology space, targeting various types of cancers, liquid tumors being the primary target, and now moving towards much more of solid tumors as well. And then in a very much more exciting field at this point in time, because of the regulatory approvals of CAR T cells, that we are seeing a lot of efforts in using stem cell therapies, because there has been close to 5-6 decades of research that has been going on around stem cell therapy. And with that in mind, we will have to see how regenerative medicine that targets various types of aging-related diseases could be treated through these stem cell products that are either genetically engineered or they have been modified in some ways to exert the necessary therapeutic effect, right? So in terms of cell tissue therapies, there are two broad classes, right? Allogenic therapies, where patients receive cells from a donor that may or may not require HLA or human antigen leukocyte matching, antigen matching, depending on the cell type. Say, for example, for various type of immune cells, of course, at this point in time, allogenic therapies are still a bit in its early stages of development. But for example, if you're using mesenchymal stromal cells, these are ideal cell products that can be used in an allogenic setting. Then for autologous therapies, which are primarily our immune cell therapy products, these are the ones where you use patients' own cells to develop and to make the therapeutic products that are given to the patients. Estella, can I just confirm my voice is clear?
Yes, you are.
Okay, thank you very much. So for the purpose of today's presentation, we will really focus on CAR T cells, right? And before I move into giving you a broader perspective of what CAR T cells are, I just wanted to stop and give you an overview of the different therapeutic modalities that are available to treat cancer, right? Of course, the ones that we are very familiar with: surgery, chemotherapy, radiation therapy, classical ones that has been in clinical practice for many, many decades so far. Then, of course, we have targeted therapies, hormone therapy, stem cell transplant, particularly to treat hematopoietic or blood and bone marrow disorders, which has been carried out for about close to 6 decades. And in fact, as I will show in the next few slides, lays the foundation for all the advanced cell therapy work that we do today. And of course, the upcoming field of precision medicine. Cell-based therapies are primarily under the category of immunotherapy, right? And in cancer immunotherapy, we see four broad classes of therapeutics. You have adoptive immunotherapy, part of which is actually CAR T cells. And then there are other forms of immunotherapy, adoptive immunotherapy as well. Then you have the use of cytokines to treat as well, and then monoclonal antibodies and subsequently cancer vaccines as well. So for today's presentation, we'll be really focusing on the adoptive immunotherapy and looking at the subclass of that, which is CAR T cells. Okay, just now I mentioned that stem cell transplant is the backbone structure from a clinical perspective that you need to have in place to implement any sort of cell tissue and gene therapy effort in the clinic and to bring it to patients, right? This is extremely essential because the knowledge that we have garnered, the technologies that we have invented, and the clinical processes, laboratory processes that we have put in place is really completely reusable by the cell tissue gene therapy ecosystem and product developers to bring their products to patients. So for example, hematopoietic stem cell transplant was first discovered in the 1960s. And since then, this field has evolved greatly through the invention of human leukocyte antigen for better matching of donor and recipient cells, through the use of immunosuppressive drugs that has enabled the control of graft versus host disease that we see post-transplants, as well as the development of various anti-infectious disease drugs that has been critical in bringing together patients, and of course the various types of chemotherapies that have been put in place to enable hematopoietic stem cell transplant. Many of these protocols, many of these products are highly relevant in bringing cell therapy to patients, right? I just wanted to stop here and also highlight, since this talk is hosted by ACTRIS, ACTRIS being a national platform, right, that is mandated to facilitate discovery process development as well as clinical scale manufacturing of cell tissue and gene therapy products. I just wanted to highlight here that Singapore, in fact, for the past 3 decades or so, has been the regional referral center for hematopoietic stem cell transplants. And later in my presentation, I'll show you and talk about more of the clinical sites that we have. But I just want to highlight here that we have been performing close to about, as an island, close to about 300 over transplants per annum. And given the size of the population that we cater to and the patients who can access these therapies, this is in fact a very high number. And we are possible to do this because we have internationally recognized and accredited clinical care hospitals and services, processing laboratories, donor registries, cell banks, and in fact, we are just not performing conventional types of hematopoietic stem cell transplant, but have ventured into performing and even routinizing very advanced form of therapies across our hospitals. Okay, what are CAR T cells? Very simply put together, these in most cases are your patient's own cells that are taken out of the patient's body. And I'll talk more about that in the manufacturing segment of my presentation. A CAR gene is introduced through a plasmid and a viral vector. And these CAR genes then are expressed, or chimeric antigen receptors are expressed that target specific markers on cancer cells and goes and binds to these cells and kills the cell, right? So of course, this CAR construct is one of the key mechanism of action, or the active pharmaceutical ingredient for this CAR T cell product, right? Over the years, we have various generations of CAR T cells that have came out. In fact, early studies for CAR T cells began in the 1990s. And many of these work were initially funded by philanthropic efforts that highlights how important that is for such therapies and innovations to be supported through philanthropy, through research grants, to really bring it to patients. So what you see here is that in the first generation, we had very crude form of CAR Ts, which was further refined. And the top part, which is the tumor cell recognition part, is shown here. And then, of course, you have your post membrane area where you have the activation, proliferation, and killing activities that takes care of the various other mechanism of action that's necessary for CAR T cell to exert its effect, right? CAR T cell signal amplification is based on synthetic biology. It has avidity control. CAR target surfaces, CAR only target surface structures such as proteins and glycans. Is MHC independent recognition of tumor targets. At least it will have decade-long presence. The initial studies or the initial infusion of CAR T cell to a patient has only happened in the early 2010s, around to 2011 and 12. So we are still monitoring in various regulatory authorities and hospitals. There are serial killers of tumor cells. And of course, one of the key side effects of CAR T cell is cytokine release syndrome. That again, I will touch base in the later part of the presentation. Okay, so just now I mentioned that we have the domain where you have the co-stimulatory factors in the last slide, which enables the CAR T cells to proliferate and exert its effect. So there are various mechanisms or strategies for this co-stimulatory support, right? This again, I will just go back one slide. I'm talking about this part here. Physiological co-stimulation, where ligand display by artificial antigen cells are done, antigen presenting cells are done. Then you have auto-trans co-stimulation by T cells expressing co-stimulatory ligands, as you have seen in the last slide. Then you have a redirected co-stimulation mediated by an antigen-specific chimeric co-stimulatory receptor, as shown here. And then, of course, the last kind that is being developed for the next generation CAR T cells are embedded co-stimulation provided by second or third generation CARs. And in fact, there are fourth generation of CARs that are coming in place that gives you better control on how the cells behave inside the patient's body, giving greater safety and efficacious outcomes. Just earlier in the presentation, I mentioned that a cornerstone for any hospital to implement CAR T cell services is to have hematopoietic stem cell transplant. And this recent publication really tries to exemplify the similarities and differences between, for example, autologous hematopoietic stem cell transplant and a CAR T, as well as an autologous CAR T cell therapy. So on the top, what you're seeing is the process, a clinical process, end-to-end clinical process from cell collection all the way bringing back the cell to the patients and doing acute follow-up to long-term follow-up that is necessary is highlighted here, right? So on the top is autologous hematopoietic stem cell transplant. On the bottom are your CAR T cell therapies, right? Over here, what you see is that during an autologous hematopoietic stem cell transplant, the cells that we are collecting are stem cells. These are CD34 positive stem cells. If the patient is going for a CAR T therapy, then we are collecting the peripheral blood mononucleated cells that has the immune cells, right? Then the cells, similar in both the steps, are sent to a cell processing center. In most cases, for hematopoietic stem cell transplant, the processing at this point in time is done on site, whereas for CAR T cells, given the complexity of manufacturing that is needed and the various steps that are needed, these have to be, in many cases, sent to specialized cGMP facilities. Again, that will be touched upon in the later part of the presentation. Then, as the cells are being prepared, the patient needs to be prepared to get these cells back for having the therapeutic effect, right? During hematopoietic stem cell transplant, majority of the patients will receive high-dose chemotherapy that will provide myeloablation, whereas for CAR T cells, we primarily give the patients lymphodepletion chemotherapies that will get rid of the excess immune cells, giving the space for the CAR T cells to come and exert the effect. Then, once the cell therapy products are ready, so one thing to note is that for hematopoietic stem cell transplant, the products can be prepared within a day or two, whereas for CAR T cells, we do need a long manufacturing time at this point in time that can range from anything between 2 to 3 weeks. The cell therapy products are brought back to the patient, is shipped back to the clinical site, and then it is given to the patient where a transplant team would provide acute phase care and then subsequent to that long-term care that's necessary to ensure that the CAR T cells persist, that they provide the effect that has been intended to do so, and that there is remission of disease, right? So how does a CAR T cell work when it has been given to a patient, right? So CAR T cells are infused into a patient's bloodstream, very much so similar way that hematopoietic stem cell transplant is done, right? The cells, once it enters the patient's body, undergo extensive proliferation, and then through the blood vessel, it will travel to the site for where the tumor cells are. It will travel to the tumor cells. And then, as I have shown earlier, by binding between the antigen and the receptor, we will see an effect of the CAR T cells that will cause the death of cancer cell and as well at the same time release various antigens. These new antigens are then presented to antigen presenting cells that will subsequently go on to prime endogenous T cells and activate them for further killing action. Right. So why is there so much of excitement with CAR-T is really summarized on the table that you see on the right-hand side. These are very early phase clinical trial studies that have been reported and rather pivotal studies that has set forth the approvals for the various CAR-T cell products that we see in the market right now. So first of all, CD19, that was a target for various B-cell diseases, acute lymphoblastic leukemia, diffuse large B-cell lymphoma, and so on and so forth, that has been the primary target. So one thing to note here is that the patients who came for these trials had failed several lines of therapies, whether that was an autologous transplant, whether that was high-dose chemotherapy, whether those were targeted therapies, for example, through monoclonal antibodies, and so on and so forth. So these patients were highly relapsed and refractory, right? So, and then the response rate that you see with these patients receiving CAR-T is amazing. From 80% onwards that you have a response rate, 60% to 70%. And these were patients that did not have very good prognosis and survival if the CAR-Ts were not offered to them. So at this point in time, for leukemia, lymphoma, one of the key targets is CD19. And for multiple myeloma, refractory multiple myeloma is anti-BCMA. So later on the presentation, again, I will speak about some of the other targets that are being developed. With that, just wanted to bring your attention to the number of clinical trials that's happening across the globe. This figure is from a 2018 publication, but the status quo has been maintained, where we see that at least over 1,000 clinical trials are registered and taking place through major regulatory authorities such as FDA, EMA, and most recently through efforts through HSCT that I will talk about in the later part again. And some of the dominant, very, very prominent CAR T cells that have been approved, for example, in FDA, Kymriah, and as well as various other products that are targeting, again, B-cell diseases. So one thing to note that amongst the various cell therapy trials that are taking place, CAR T cells really dominates majority of the trials, and that has been an area of steep growth over the past few years. In terms of the targets that are investigated through these various trials, right, are actually highlighted here, as you can see. For hematological malignancies, we do see markers such as CD19, 20, 22, 30, 33, CD1, 2, 3, and so on and so forth. Of course, BCMA is the primary target for multiple myeloma. And when it comes to solid tumors, there's also significant efforts that are taking place. And as you can see on the left-hand side here, as well as on the right-hand side here, the various targets that are being explored for CAR T cells, and the diseases that are being targeted through these various targets right now. So we do see a growing number of trials in the solid tumor space, but there are, of course, some limitations when you're applying CAR T cell for solid tumors. Okay, one of the common adverse effect of CAR-T, again, from a researcher perspective, trying to understand the basics of CAR-T as you try to develop these products, is definitely of cytokine release syndrome, right? What is basically a cytokine release syndrome? It is when a CAR-T engages surrogate antigens, cytokines and chemokines are released, right? As you can see on this diagram here. These innate immune system becomes activated, release other soluble factors, right? And these cytokine increases permeability to soluble mediators and permit increased trafficking of the CAR T cells into the CNS, where you will get various neurotoxicities as well as various other effects relating to cytokine release syndrome, right? One way to overcome this is to use various antibodies and so on and so forth. For example, tocilizumab, which is heavily used to treat side effects of CAR T cells. Although this field is very exciting, growing, but there are certain limitations to CAR T cells, right? There are patients who experience, even after having initial response, high response rate to the therapy, they do progress with the disease. There's a CAR failure, which in many cases could be many things associated to it. You could not harvest enough cells, the CAR T cells did not expand enough during the manufacturing process, or the CAR T cells did not persist well inside the body. Antigen modulation, which in which case you do see that, for example, the initial disease had overexpression of CD19, but the relapse disease had overexpression of some other marker. For example, in the B-cell diseases now, there are lots of studies coming up showing that CD19 CD19 and 20 probably has to be targeted together. Of course, as I mentioned, CAR T toxicities, and there are a lot of unmet need with regards to CAR T cells in the solid tumor space, primarily because of the reasons that you see highlighted on the right-hand side. Heterogeneity, cells of solid tumor do not all present the same mix of antigens on the surface that we usually see with liquid tumors. And of course, solid tumors has a microenvironment that has been known to provide immunosuppressive effect to immune cells. So therefore, the effectivity of CAR T cell does drop in those microenvironment, and how to overcome this. And of course, for liquid tumors, one way to deliver CAR T cells is intravenous infusion. However, for solid tumors, intravenous infusion may not be the most ideal route for delivery of CAR T cells, and therefore new technologies or approaches towards delivery of these cells needs to be explored and developed. Right. So some of these challenges, what are ongoing efforts in this entire field to improve the safety and efficacy of CAR T cells? Of course, one of the key parameters for safety is to predict individual patient risk for the complications that arises, for example, cytokine release syndrome and neurotoxicity. That's something that we need to address. Having switchable CARs that are one of an example for which I will highlight in the next few slides would be again something desirable, because that will enable us to control its therapeutic effect as well as its side effect. Suicide systems where CAR T cells can self-destruct itself after it has exerted its desired effect to minimize other side effects. For example, in CD19 CAR T cell, we see prolonged B cell aplasia, right? Then, of course, exploring transient expression systems, again, that can control the side effects of CAR T, affinity-tuned CAR T cells, lower affinity for specific antigens that could be targeting healthy cells, or we can even talk about tandem CARs that are taking place right now, where two CAR cytoplasmic signaling domains are added onto two CAR molecules, right? To increase efficacy of CAR T cells, yes, we have astounding response rate for relapsed refractory patients, but to move up CAR T to frontline therapies, we have to have improved efficacy. So whether combination of CAR T cells with the immune checkpoint inhibitors or kinase inhibitors is one area that is being explored through various clinical trials. Armored CARs, specifically for solid tumors, where they would co-express pro-inflammatory factors such as IL-12 or IL-15 for increased persistence and proliferation within the immunosuppressive domain of solid tumors. Co-expression of two CARs that target different antigens, for example, CD19 and 22. And I'll illustrate an example shortly on this. And incorporating human-derived rather than mouse-derived scFv domains into CARs. The initial CARs, some of them were mouse-derived, but now companies and developers are moving away from mouse-derived scFvs to human-derived to increase persistence and as well as avoid various immune responses that we see in patients. Right. So this brings me to the next part of my presentation, recent scientific discoveries. Really, there is a growing number of publications every year, at least 1,000 over publications that we see for the past 2 to 3 years with regards to CAR T cells, almost coming to on an annual basis. So just to highlight a few, as I mentioned, for the primary target of CAR T cell was CD19 positive B cell diseases, right? So this study, which reports a phase 1 trial of testing a new target, CD22, in children along with adults, including 17 who had previously been treated with CD19 targeted immunotherapy, some of whom they received CAR T cells. This is just one of the studies. There was again another recent study for which I do not really have the time to highlight. What it basically shows, most importantly, is that when you combine a bispecific CAR, which has CD19 and CD22, you see that the disease has been controlled much better than instead of giving just one specific CAR. So this, again, these studies demonstrate that bispecific CARs, very much similar to bispecific antibodies, do have relevance and importance in the next stages of CAR T development, right? Next study, again, very similar. Over here, we are not talking about bispecific CAR, but giving two types of CAR T cells. This is in a relapse refractory multiple myeloma setting, where studies have shown that multiple myeloma patients, on top of the anti-B cell maturation antigen, which is the BCMA, the primary target for CAR T cells for multiple myeloma, that there is still a group of patients who do have less differentiation myeloma clones expressing CD19. So giving these patients, infusion of humanized anti-CD19 CAR T as well as a murine anti-BCMA CAR T yielded better clinical outcomes, right? The next study, as I mentioned earlier, tyrosine kinase inhibitor, dasatinib, acts as a pharmacological on and off switch for CAR T cells. So why is this beneficial to have on and off switches for CAR T cells? Because that helps us to regulate the effect of the CAR T cell as well as its negative side effects that we see in patients, for example, CRS and neurotoxicity, right? In this preclinical study, this was purely a preclinical study which showed that dasatinib interferes with the lymphocyte-specific protein tyrosine kinase. And through this interaction, that it could control the ability of the CAR T cells to be activated or be deactivated. And this graph, which is sort of like a co-culture system, in vitro system, but of course the study has gone on to do significant in vivo studies, shows that enriched CD19 positive CAR T cells were co-cultured with the CD19 positive target cell in presence and absence of dasatinib, and you can see that the specific lysis was severely impacted off the target cells when the dosage of dasatinib was increased, showing that this is a mechanism through which CAR T cells can be switched on and off, right? Further studies were done to illustrate that as you switch on and off in an animal model, that you can see the effect where the tumor size can be measured was growing or not growing at the point in time when the CAR T cells were activated. This is a very interesting study, again, a purely animal study, and this speaks to about the challenges when we have to look at targets for CAR T cells. CD19 was a very good target because for developing CAR Ts. But say, for example, when you're looking at myeloid diseases where the leukemic cells or the cancer cells as well as healthy cells have expression of the same marker, the ability to use CAR T cells becomes limited, because once you introduce a CAR T cell that targets a marker that is widely expressed on healthy cells, that means that those healthy cells will also be killed by those CAR T cells. So this study was in fact very interesting in trying to develop or propose the more way forward for developing CAR T cells for acute myeloid leukemia. In acute myeloid leukemia, CD33 is widely expressed, but CD33 is also widely expressed on all myeloid cells across our body, including other cell types as well, right? What this study did was they did show through appropriate animal model and in vitro testing that CD33 is not required, say, for example, human myeloid development and function. So in that case, what they did was to get rid of CD33 from human hematopoietic stem cells. They transplanted these cells into mice. And then, of course, they also engrafted the cells or grafted AML cells onto the mice. And then after a certain point, the hematopoiesis was recreated in the mouse model. The tumor cells grew within the same mouse model. Anti-CD33 CAR T was given, and that showed that you could kill the acute myeloid leukemia cells without impacting the human hematopoietic stem cells that were given with CD33 knockout. So this is a very interesting study and in an animal model, but what it means for human studies in future is that you can have healthy donor where you take out the human hematopoietic stem cells, you do gene editing to get rid of these CD33 marker, because these preliminary preclinical studies does show, which requires definitely further validation and further verification before they can be tried out in humans, that these cells can be given to patients, and simultaneously at the same time you could be culturing and expanding CAR T cells that target CD33, and then you give to the patient at the same time, enabling you to recreate both, enabling you to perform both a hematopoietic stem cell transplant as well as a CAR T cell therapy to treat acute myeloid leukemia, which in fact has been a major disease that has not had any recent breakthroughs to overcome the current clinical outcomes that we see. Another challenge for CAR T cell manufacturing is T cell exhaustion. These two studies are very basic studies. I just wanted to highlight that T cell exhaustion is a real problem for CAR T cell manufacturing, especially when you're collecting T cells from patients who have been heavily pre-treated through various other modalities such as chemotherapy, targeted therapy. And when you have T cell exhaustion, you are creating a product that is suboptimal, that has poor specifications, or maybe out of specifications products, as regulators would like to say. And these cells, when given to patients, may exert suboptimal effect. So these two studies, although not specific to CAR T cells, but specific to T cell biology, that showed that T cell exhaustion could be correlated to tox expression, right? So one way to move forward and look at these kind of basic data and to translate them into clinical manufacturing processes of products is to see, could we generate CAR T cells that were further engineered to lack tox that would enable these cells to grow better, be lesser exhausted at the end of the expansion process. T cells get exhausted in vitro cultures because we push them with cytokines, we push them with activators to ensure that they survive in the culture and that they grow to numbers that are of relevance for giving back to patients. I just wanted to deviate away a little bit from immuno-oncology applications for CAR T cells and bring your attention to this very recent study that was published. And really, this is at the interface of CAR T cell or immuno-oncology applications, as well as regenerative medicine, which I spoke about earlier in the presentation, that really comes at that edge of these two therapies coming together. So this study, again, is a very upstream animal study, which was very interesting to see that you are using senolytic, that these guys were using senolytic CAR T cells to reverse senescence-associated pathology. So allow me to explain a bit, right? Senescent cells exhibit a response called senescence-associated secretory phenotype, okay, SASP. This is associated with the release of various molecules that attract immune cells, and then these cells would kill these cells, right? But in many cases, senescent cells start to accumulate, and they give rise to various diseases in our body, including cancer, including fibrosis, right? In this study, what they showed was that they had identified a protein called UPAR that is expressed on the surface of senescent cells, and CAR T cells were engineered to express an antigen, chimeric antigen receptor against this protein, which, when these were given to the in vitro and were tested out in the in vitro and in vivo model, could show that they could kill senescent cells. So just a bit of, trying to address the various challenges. Of course, all these studies that have illustrated to you are all autologous therapies. That means you manufacture one batch for one patient, right? But that when it comes to scalability, when it comes to manufacturing cost-effective and cost-efficient therapies is really very challenging, right? So there are lots of effort ongoing. I have not summarized all the clinical trials that are taking place, but there are a handful that are taking place, for example, in territories like US and China, that are looking at using allogeneic CAR T cells. So in this case, instead of taking the patient's disease cells who have been pre-treated, heavily pre-treated, the cells may have been pre-exposed to different types of chemotherapy, targeted therapy, radiation therapy. What this entire domain of expertise and group of people are trying to do is to collect healthy donor T cells, use viral vector to make CAR T cells, but at the same time they're performing. So this is gene engineering to introduce the CAR into the cells, but at the same time they're also performing gene editing to get rid of certain things that would make a CAR T cell from an autologous product to an allogeneic product, right? What do they do is there is gene editing done on the TRAC track, T cell receptor alpha constant, or TRBC, T cell receptor beta constant genes that encodes for the TCR alpha or beta chain that you see here, right? Knockout of either locus will disrupt the TCR alpha beta pairing and hence eliminate surface expression of the TCR CD3 complex, right? So this entire complex will disappear. An absence of TCR does elevate alloreactivity or autoreactivity or alloreactivity of the engineered T cells. And to perform these, the gene editing tools that are widely used are Zinc finger talons and of recently CRISPR becoming really, really prevalent in this field at this point in time. So after you do gene engineering, you have done gene editing, the cells go on for amplification, and then after that, you do a final step of filtration to remove cells for whom the TCR was not disrupted as a result of the inefficiency of the gene editing process. Then you are having a CAR T cell product that can be used in an allogenic setting. And of course, with that concept and knowledge, various other things are being knocked out or knocked in TC using the TCR, this platform that I have shown you to increase persistence, environment potency, and homing of CAR T cells targeting these various markers that are listed here. Okay, nonetheless, this is my last slide on the studies that I wanted to highlight. One thing to bring forth, and this is something very important for especially for researchers who are out there, product developers who are out there, that as we innovate different products, trying to overcome the hurdles or challenges of CAR T cell, it is very important for us to keep in mind that these cellular therapies that we are developing must be safe and efficacious, and for that various regulatory mechanisms have been put in place. And this study is really a reminder of what it means to exercise different types of safety mechanisms to ensure that the product is safe, for which manufacturing must be done in a very controlled environment. So this study was really very interesting for many of us to read who have been following this field, right? Is that induction of resistance to chimeric antigen receptor by transduction of a single leukemic B cell. So during the manufacturing process, the CAR gene, right, was unintentionally introduced to a single leukemia B cell that was in the starting material, and that in itself became the source for relapse and new disease in the patient. So something for us to look out for, as we are also excited about this growing field, but something to be again aware of, that safety, quality management system, and putting in the appropriate regulatory mechanisms to ensure that the products that are given to patients are safe is of the utmost importance. Okay, with that, I'll move towards manufacturing processes, and I do think that I'm running out of time, and to enable us to have some more time with regards to our Q&A session, right? Manufacturing of CAR T cells versus biologics, right? Just wanted to give you an overview. On the left-hand side, you see a conventional drug and biologic that is manufactured in a centralized facility, mostly dominated by the pharmaceutical industry, where one product lot gives rise to many doses for many patients, right? But whereas for cell tissue and gene therapy products, we are seeing that there is, especially in the autologous therapy space, that there is much room and space for decentralized manufacturing. Therefore, there are lots of challenges that comes along with it, from process innovation to equipments, and even being the site of manufacturing becoming closer to the hospital rather than these centralized models that we were so used to over the past many years. And in fact, the clinical trials around cell therapies are significantly different compared to these small molecules and biologic studies that we are so used to, right? In terms of manufacturing, there are various challenges that we have to overcome. Sorry, I just can't remove this. So, okay, so for example, you need multiple disciplines, multiple technologies are needed to manufacture CAR T cells. Over here, as you can see, genetic engineering, target selection, regulatory compliance, cell manufacturing science, synthetic receptor design, as well as tumor biology all play a key role when you're trying to bring together a CAR T cell manufacturing process, right, for patient use. Just to give you an overview, of how the manufacturing process looks like. You collect cells. In some cases, you start with fresh cells. Some cases, you start with frozen cells. In that case, you have to thaw cells. You have to do debulking of the cells or enrichment of the cells, for which there are various technologies out there, clinically relevant technologies out there. Then you do cell activation, viral delivery, cell expansion, harvest, debedding, and going on to washing and concentration formulation, and then bringing the product to the patient. The whole process, what you need to watch out for is that you have sufficient expansion of the target cells that has the target gene inside it, such that you receive a therapeutic dosage that is of relevance for use in the clinic. For example, for most CAR T cell studies right now, patients are dosed at least 1 million CAR-positive viable cells per kg or per kilogram of their body weight. Right. In terms of manufacturing, again, one thing to take note of, that manufacturing is of utmost importance because manufacturing strongly contributes to the cost of these therapies. One thing that I do not have time to touch upon, but you can talk about more on this during the panel discussion, that the cost of these therapies have really taken aback many of the reimbursement mechanisms that have been classically been put in place. So in terms of manufacturing, as you can see, it is one of the biggest cost driver of cell therapy cost, right? Many things that contribute to it: viral vectors, cell selection, reagents, growth factors, activators, and so on and so forth, on top of the usage of the facility, the processing, the labor, and the quality control system that you need to put in place to ensure that these products are safe and efficacious. One way to overcome cost-related barriers with cell therapy manufacturing is to have automation incorporated in your processes, right? And this study, which was done, has shown justifiably that when you bring in place automated closed processes, have reagents that are regulatory compliant, so on and so forth, that your cost can reduce by up to 37% for for manufacturing a single batch of CAR T cells. There are various type of platforms and equipments and automation platforms that are out there that you can choose to use. You can use a modular-based platform where each equipment performs. And again, these are just for exemplification and educational purposes, just a gentle reminder to my audience that, in a modular system, one machine does one step of the manufacturing process, for example, cell selection, expansion, harvest, and so on and so forth. However, there are also very new technologies that have came in place that are single platform technologies that can do the entire process in a closed system, in some cases not requiring a grade B room that we are so used to having, and bringing down further the cost of manufacturing. So when you're choosing between a modular platform or a single platform, various things to take into consideration: cost, scalability, efficiency, the stage of the clinical trial, flexibility, and processes required to incorporate changes. Once the CAR T cells are manufactured, again, it is essential for us to characterize these cells to ensure that they have the right characteristics. So, right characteristics for infusion into patients. So, for example, you need to do tests that would establish safety, tests that would establish purity, identity, potency for these products, and the various types of tests that are usually done in a clinical setting is shown here, right? So these are various things that you need to look out for. And again, release test is another activity that does significantly contribute to cost for cell therapy manufacturing and cell therapy products, right? One reminder to my audience who are mostly from the research community that selecting appropriate reagents that have the right regulatory status are essential as you think about CAR T cell development, right? Something that is, again, can be the content of an entire 40 minutes of lecture is to say that clinical reagents, if can be used, are of course preferred by our regulators. However, not all reagents have clinical grade available in the market. Therefore, when you're using new reagents, make sure that different types of documentations are available to support the safety of these starting materials and the characteristics of these starting materials. And again, implementing a qualification program for your ancillary materials is essential when you're manufacturing to CAR T cells. Okay. So with that, I'll spend the next 3 to 5 minutes, and to close off this presentation is to talk about Singapore's growing cell tissue and gene therapy ecosystem, right? We have lots of exciting activities taking place in US and Europe, but in fact, in Singapore, we are also having a lot of exciting activities taking place now. Earlier this month, which I will show again, HSA, our regulatory authority, rolled out the regulatory framework for cell tissue gene therapy products. Commercial product was granted approval as well. And then there are lots of newspaper articles that you may come across and have seen how innovative cell therapies are given to patients across our hospitals who are who has greatly benefited from these therapies, right? Again, a gentle reminder that we are actually the regional referral center for hematopoietic stem cell transplants. So all our hospitals that you see listed here, and who may not be listed here due to sheer lack of space, is really at the forefront of performing hematopoietic stem cell transplant. That means they have the clinical infrastructure, the clinical team, as well as the clinical workflow processes to immediately implement CAR T cell therapies. And that will be again demonstrated later on, right? On top of our clinical infrastructure, we do have cell registries, cell banks, and we are developing further cell banks and registries that will enable us to manufacture CAR T cells faster and at a cheaper rate in the near future. There are a number of government-funded R&D efforts that are taking place across all the major academic universities, research institutes, and academic medical centers. This is just a snapshot of the various activities that are taking place in our various organizations. Again, due to limitation of space and time, I have not been able to include everybody. So please do bear in mind that this is just a snapshot of what we have captured through the various activities. Something to take note of: about 2 years ago, NRF, National Research Foundation, and including other funding agencies such as BMRC, gave out $80 million worth of grants that to support manufacturing processes, innovations, as well as development of various types of cell therapies that are now being developed in our various labs, right? Okay, I wanted to pause here and give you guys, to many of you who may not have had a chance to review HSA's recent website. Earlier this month, on 1st of March, HSA has rolled out the regulatory guidance and framework for cell tissue and gene therapies, right? You can go to their website that I have listed here, www.hsa.gov.sg/ctgtp to find out more, but I will try my best to cover the key points on this 1 or 2 slides that I've developed for this purpose of this presentation. So CTGTPs, as per HSA's guidance, have been classified based on their risk. Key regulatory controls have been put in place to regulate the various activities pertaining to CTGTPs. In fact, we are the first, to the best of our knowledge, we are the first ASEAN country to implement a comprehensive CTGTP regulatory framework, right? And this framework is definitely timely, seeing the number of activities that we see in our ecosystem. It has taken a very pragmatic approach because this is a very fast evolving field, progressive, and in alignment with other regulatory authorities such as US FDA and EMA where we see a number of CAR T cells and cell therapy products being approved, right? And earlier this month, again, post implementation of these regulatory guidelines, there was a first approval for a commercial product that was first in Southeast Asia by HSA, right? If you want to know more or get in touch with them, please do contact them at this email address that's listed here. Further to that, I just wanted to, touch base briefly on the risk-based classification of CTGTPs that has been defined for the general audience, right? There's a class 1 CTGTP and a class 2 CTGTP. Let's focus on class 1 CTGTP, which are minimally manipulated. This refers to the techniques that you perform in the manufacturing process. Basically, minimal manipulation has definition of certain things that are within the scope. You can find it in the documents again that can be found here. These products are for homologous usage and must not be combined with therapeutic products or medical devices, right? What are class 2 CTGTPs? Class 2 CTGTPs are those that do not qualify to be class 1 CTGTPs. In most cases, these are more than minimally manipulated and for homologous usage. And I just wanted to, highlight to many of our audience, again, in the research community that, as you embark on cell tissue gene therapy research work, it's extremely important that you understand the regulatory framework as you take part in discovery work, right? And CTGTP being a very evolving field, there are actually quite a bit of fragmentation with regards to the regulatory guidelines that have been put in place by the various regulatory authorities. And in fact, earlier this year, I personally had attended this course, which was on, a comprehensive course on global regulation of CTGTPs, that's been rolled out by Duke and US Medical School. And in fact, I really found this useful, and I encourage researchers out there, product developers, to look at this and to be able to understand the various regulations and compare them across the board to understand, really how does product development work? Because a lot of the innovation for cell therapies is taking place in laboratories, in academic medical centers, unlike small molecules and biologics that have been very much centered in pharmaceutical companies. And I wanted to just end off the presentation by giving you a brief overview of what ACTRIS is, as I'm a part of this team that's led by Dr. Tan Lip Kun. Just wanted to speak about our organization, our core capabilities briefly. We are going to be a centralized manufacturing and process development facility serving a wide range of clientele across Singapore. Our infrastructure, basic equipments, specifically our infrastructure design has been reviewed by FDA, HSA, and we are bringing together various capabilities, studying other similar centers across in US and Europe to ensure that we can provide end-to-end clinical process development and manufacturing capabilities to expedite the development of cell tissue and gene therapy products over the next 5 to 10 years. Just a brief overview. I seek your understanding that this is a non-exhaustive list, but this is a map of Singapore trying to show the ecosystem for cell tissue and gene therapy that has been growing. In fact, from an ACTRIS's point of view, where we see ourselves as the pivotal center sitting at the interface of the public and private sector, trying to moderate that growth of cell therapy manufacturing processes, that key players are all over the island, from startup companies, industry, ancillary industry, our hospitals that are bringing these therapies to patients, research centers such as A-STAR doing groundbreaking work in this space, as well as our universities, then our ministries and regulatory authorities or stat boards that are coming together, including funding agencies, other manufacturing sites, and as well as investment. I seek your understanding. Again, this is purely for educational purposes. Last but not the least, this is a slide that we put together for ACTRIS in most of our roadshows, talk and studies. just to presentations, just to show that, with the introduction of ACTRIS and this entire ecosystem that you see, how are we going to interact in developing products from an early stage of discovery and bringing it all the way to large scale manufacturing for regional, of course, local, regional, and global distribution, right? So we see that there are various partners who play various activities in this entire product developmental pathway: academic universities, research institutes, medical centers, focusing on discovery, going all the way up to preclinical animal studies, when organizations like ourselves and other organizations such as A-STAR, BTI coming together to bridge that gap for translation that needs to do so, to bring a product from preclinical animal stage to small mid-scale manufacturing that supports early phase clinical trials that are so much critical in generating safety and efficacious data. And these products then has to move on to large scale manufacturing once approved by regulatory authorities for, again, as I mentioned, for global distribution. And all of these activities, right, are taking place with the recent introduction of HSA CTGTP regulatory framework that ensures the development and the implementation of a robust clinical quality management system to support the entire ecosystem. As this is an education webinar series, I just wanted to highlight, right, that as I was preparing this slide deck, that over the past 6 months to about a year, actually a number of webinars, seminars, and conferences have been rolled out by various organizations within Singapore that are trying to create awareness, trying to increase the knowledge and the technical skills, bringing together these webinars and conferences. For example, Stem Cell Society Singapore has been, brilliant in running large-scale conferences with other international organizations. We have our Singapore Cell Therapy Advanced Manufacturing Program that I just now spoke about, our flagship R&D program that was funded about 2 years ago, that are also running various webinar series focusing at the R&D level. Then we have a recently established Singapore Duke and Cell Therapy Center that again, runs webinars and working towards developing conferences as well. And of course, ACTRIS's webinar and other upcoming education initiatives that will be coming in place to really address the skill set and as well as the growth of skilled manpower in this area, hopefully in partnership with academic universities over the next year or so. Do feel free to reach out to our education team at education@actris.cris.sg if you would like to find out more about these initiatives, and feel free to attend some of these webinars that are hosted by various organizations. ACTRIS's webinar runs once a month. So with that, I would like to thank everyone. Apologies that I overran a little bit.
Okay, thanks, Dr. Bari, for this very informative sharing. Now let's also welcome Dr. Tan to join us as the panel. I think there are quite a number of questions that have already been posted at the Q&A section. Hand this time over to Dr. Tan as well as Dr. Bari.
Dr. Tan.
Yeah, hi, Sudipto. Thanks very much, Stella. I think there are a few questions come in. Sudipto, shall we go through them?
Yes, Dr. Tan. So the first one that I see on my screen is, I'll just read out to you so you could probably answer on this point. What is the evidence for secondary killing by the endogenous T cells after CAR T cell treatment? What is the evidence that is based on new antigen release where presumably the new antigen existed prior to application of CAR T cells? Would you like to take this question, Dr. Tan?
I think for this question, I will have to refer to you. I'm not actually quite sure, but we can also, of course, get back to you on the answers later on via email. All these questions will be answered at the end of the talk, and we will send to the respective people that have sent them.
Yes, so yes, agree with Dr. Tan. Very briefly, yes, there is speculation that does endogenous T cells does, if convert that secondary killing effect. But as you know, that there's a lot more animal studies and as well as human studies that is necessary to answer, this specific question. So I'll move on to the next question. For CAR T cell cryopreservation, does the field utilize serum containing or serum-free formulation for long-term storage?
So currently we've been using DMSO to freeze them down, but of course there are still serum-based formulas that we are using, but we are slowly moving towards non-serum methods.
Right. What is the progress to commercialization, example through Singapore-based spin-out and startup companies?
Sudip, do you want to take this one?
Yeah, sure, Dr. Tan. At this point in time, as I had earlier mentioned today, we have only seen the commercial approval for one of the product earlier this month by HSA after they have implemented their, cell tissue and gene therapy regulatory framework. Right now, what is the framework or what would be the pathway? There are a number of startup companies that I had shown earlier in the map. They are in the early stages of development. Some of them are carrying out clinical trials. Some have completed, in fact, multi-center clinical trials, as we know. We yet to see an approval for a product that was completely homegrown. So there's something for us to watch out for. But again, going back to the regulatory page that has been, introduced by HSA, introducing the entire regulatory framework does provide a complete, landscape of how manufacturing, movement of the product, infusion, follow-up, clinical trial must take place. So that's something to watch out for. Yes.
Yes, Sudip is right. And in fact, on the HSC website, there are information on this. I mean, for companies so far, we've not seen yet commercialization of homegrown products. There are products which are, of course, going through trials. A couple of them are being managed in NUH itself, but homegrown products itself have not yet been commercialized within Singapore domain. But the systems of how to commercialize has been put in place.
Right. So do ACTRIS provide consultation to help small biotech company develop closed system processes? Yes.
The answer is yes.
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 I think if you look at, I think Sudip, we have a slide earlier on. ACTRIS basically one of the main functions is to actually help provide this kind of service to biotechs, either in Singapore or out of Singapore, to develop their protocols and help them with manufacturing as well.
So yes, just to illustrate further with our slide, right, that yes, we do have the capabilities that we're bringing in. We are bringing in various commercially available platform technologies for manufacturing, as well as evaluating new technologies that are being developed by our local research teams, right? We're working closely, and that's our planned way forward, right? That we will have all these capabilities within the facility once it's fully operational. Okay, so I'll move on to the next question. Stop share. What would be your recommendation for device manufacturers? How can we support better? Is it on product portfolio or rather support earlier stage initiatives? Dr. Dan, would you like to take that?
I think for devices or equipments, what we have been doing is doing comparative side-by-side studies before they get registered as devices, registered with devices. So we are working with several groups in A*STAR to do this on their own prototypes before they become proper equipments for usage.
Right. Any further points to add? I think the device or equipment providers and reagent providers would probably have to closely work, with just from the manufacturing side, but also create awareness of the various manufacturing platforms and reagents that are available to various researchers and as well as startup companies who are working in this space. So I think we are running a bit low on time. So we probably will take one or two last questions. Okay, are there groups working on allogeneic CAR-T or hematopoietic stem cells in Singapore?
Yes, there are some groups who are doing that. Of course, we are not able to reveal who they are at this point to you.
Okay, testing RCL is a key parameter in batch release of CAR-T product. It takes time to complete this test. Do you accept any new approaches, example, fast molecular assays to replace conventional method? Dr. Tan, maybe I will take this question. So yes, these again new tests similar to any implementing any clinical test in our laboratories, and as well as getting this approved by regulators, requires validation, right? So as long as we have the appropriate validation against the control test or the standard of care test that we have in place now, definitely we can look into it. But however, the final jurisdiction as to whether this test is acceptable when you submit a CMC still lies with the regulatory authorities. But of course, as a manufacturing side, our due diligence would be to carry out the necessary validation and verification work, and to do the comparative studies that Dr. Tan had mentioned earlier. Okay,
Yeah, so
maybe, yes.
Yeah, so all these have to be compared against standard recognized tests before we can change them to newer methods, and they will always have to be assessed with standard methods first.
Dr. Tan, maybe one last question. Okay, and this is more appropriate with the hematopoietic stem cell background. Is there any developed cold chain management or guideline for CAR T-cell products?
Which one is it?
The last one, is there any developed cold chain management or guideline for the CAR T-cell or cell therapy products?
Currently, this is described in the standards, but if you talk about an actual protocol available, I would say no, not at this present time. But many of the cold chain requirements are actually within the standards itself. But each lab or each facility will have to come up with their own cold chain management for now.
Okay, so maybe one last question. Again, it's a clinical question. HSA would need to proceed clinical trial in Singapore population for CD19 CAR T-cell therapy to be approved. This is a very specific question towards HSA, so maybe we encourage them to write to HSA directly. Dr. Tan, is that okay?
Yeah, I think because it's directed at HSA, maybe you could. They do have email that you can send to and ask them. In general, yes, there is need to still carry out if it's a homegrown CD19. Yes, the current Novartis trial has been completed, and Novartis is now available as a clinical product in Singapore.
So I think we are almost 5 minutes past or 7 minutes past our closing time of the seminar. So we probably will hand this time. There are some other questions that we haven't had a chance to answer. We will try to write back to you. We have taken note of your questions. So past this time, Dr. Tan, if you had any last comments, then over to Estella.
No,
thank you everyone for attending this webinar. I hope you have gotten a lot of useful information and being updated on what is going on. And if you have any more questions, feel free to email at education@actris.cris.sg. For those questions that we have not been able to answer, we'll try our best to answer and perhaps follow up with an email subsequently. That's the end of the webinar, and thank you for your attendance. I hope to see you at the next webinar. Thank you. Bye-bye.